All right, so we're now going to move on to plant growth and development. And first, we're just going to think about some basic features of plants that are different from animals. And a lot of these we've talked about before, and they're pretty straightforward and intuitive, right? Plants can't move, right? Can't go anywhere, rooted in place. Therefore, they have different ways of dealing with challenges that they encounter, and one of the ways they handle these challenges is to grow. So we're going to find that plants respond to changes in their environment by modifying size and their shape, and they do this because they're able to grow throughout their life. So this is the growth throughout their life is how they're able to continuously modify size and shape. So we talked about this before. This slide should look familiar. Plant and animal multicellularity. They both share this characteristic, and yet they don't have a common ancestor who was multicellular. So while they had to evolve similar traits in order to be considered multicellular and a complex multicellular organism at that, their organisms over time evolved different mechanisms of having those same traits. And one thing that really differentiates plants from animals is the fact that they have a cell wall. So due to that cell wall, which does have wonderful features because it provides support, it also has consequences, meaning that it can't move. And so if you can't have change in shape of a cell, then you're going to, or of the plant overall in terms of movement, then you're going to have different ways to get around the problem. And so this picture here, I'm going to talk more about in the next slide, but what you're going to note here is that cells are going to grow in length, and they're going to have these different zones, and that will allow for them to differentiate and continue to grow. But again, we're going to see that there isn't this sort of movement that you see for animals. So in animals, we saw gastrulation. The cells literally are going to move and have new shapes. We don't see that here. We see more of growth in given directions, different ways of elongating, and we'll talk more about that. So while in animals, we talk about things like gastrulation in development, right? We see this massive change in shape of the embryo as it's developing. We don't see that in plants. So in plants, we're really limited to a few basic features, and we're going to talk about cell division, cell expansion that's going to lead to growth, and then finally differentiation. And all of this is made possible because at the very tip of a lot of structures we're going to talk about, we're going to have meristems, which I'll define more in a minute. But basically meristems, just to give you a quick preview, are stem cells of the plant. And these will continuously divide and give rise to new cells that then can go under cell division. So that's the part where they divide and they grow, and they are going to make more of themselves. So cell division, mitosis continually from cells that are produced by the meristem. And then once they've gone under that division, then they're going to go through a state of expansion where they grow longer. And then finally, they're going to differentiate. And this is going to a continuing process. So then eventually, if you were to look at the part of the roots, if I can draw this, if you could look at the part of the root above this, these cells in this region would just be static. They wouldn't have any zones. They're already gone through all these stages. They're just part of the plant. But then this part down here is continuously going through this process of meristems giving rise to new cells. Those are going to go through cell division. Those are going to expand. Those are going to differentiate. And then they become part of this stuff up here, and it just keeps happening over and over. And this is really the only way that plants are able to grow because, again, they can't move. So they're just going to continuously grow in this fashion. And we see this coming from both the tips of stems as well as roots. And we're going to talk a lot more about this. We'll look at this together in class. So before we get into the details of plant growth, we're going to just go over some basics of what their structure looks like. So if you look at any stem, they all are going to have, or shoots, right, the upper portion of the plant. Oh, the dog is barking. Okay, I left in the barking dogs. I thought you'd be amused. We're now focusing on the shoots. Hopefully she won't do that again. So we're looking at this entire shoot structure, and if we look at the shoot structure, you're going to notice a few features that they're pointing out in the picture. One is you have a leaf. That's pretty obvious. The other is that you have the distance between the leaves. That is called a internode. So there's a distance between this, where this leaf branches off, and this leaf branches off. That's called the internode, and that's because the point at which the leaf attaches to the main stem, that's called the node. And all plants are going to have this alternating leaf node internode structure. And even plants that you wouldn't, they're just going to have different ways of looking at it. So for example, you might see some plants, vines, for example, if you ever had a long viney plant, they have long internodes with very large leaves. And then on the other extreme, you have a cactus. And cacti, if you look at those pokey things are actually leaves, but they're really thin and sharp because cactus are in environments where they're going to dry out. And so you don't want to have structures that would lead to more water loss. So they've changed their structure in order to prevent water loss. And leaves, again, are the main site of photosynthesis. We talked about this before. And since they are important for photosynthesis, they need to have access to the sunlight. So you might notice if you look at a plant, the leaves aren't usually all on the same side. They tend to rotate around, and that gives them more access to the sun and prevents them from being blocked because of shading from another part of the same plant. All right, well, we were talking about the meristems before, so remember that meristems are the stem cells. You can see them here at the tips, and here you can actually see that the meristems are going to appear dark, and that's because they stained the nuclei for cells that are actively dividing. So any cell that's currently going through cell division is stained pink, so you can really see all those meristems located here at the tips. Right, so you can see them here, all the tips, and that's really important to note. And then let's think about right below that, right? So that would be your zone of division, right? So these are the actively dividing cells, and then right below that, let's say in this region, then they would be elongating, and then in this region, they'd be differentiating, and then down here, they would just kind of be done differentiated. So let's think about, again, a meristem. I told you they're stem cells, and we know that stem cells have the ability to be anything, and that's why we call them totipotent cells. So totipotency, potency means the ability to be, and toti means all. So these are cells that can become anything. They're undifferentiated. They're sort of they're sort of, just born. They can be anything that they want to be. And they are ones that tend to divide repeatedly. They are actively undergoing mitosis so that they're going to give rise to more of the same type of cell. And they express certain genes that allows them to maintain this state. Over time, even though they do divide repeatedly to give rise to more of themselves, sometimes they can become specialized. Obviously, that's what happens later on. Some of these will eventually differentiate. And a special kind of cell that they can become is a more specialized stem cell. So we see that there are the totipotent stem cells, and then there are the stem cells that are specialized for certain types of production. And so we're going to see that we're going to look at shoot apical meristems, and these are ones that are located at the tips of every branch, and they can give rise to new tissue in the shoot system, but they're going to maintain the constant size so that they're able to keep producing. We'll find in the roots you also have root apical meristems. So remember, again, we have the zone of cell division. That's where the shoot apical meristems are dividing. And then just below that, you have the zone of cell elongation. And this is where you're going to have an increase in cell size right after mitosis. And these cells are no longer expressing those meristem genes. And so they don't have that totipotency they did before. They're on their way to becoming differentiated. We're going to note here that, again, cells can't move. So in order to change their shape, they tend to grow in a polarized fashion. So we don't talk about that too much in this class, but sometimes you might say like a cell will grow wider or a cell will grow longer, and that allows it to change shape even though it can't really move, because animal cells move to change shape, plant cells can't move. So here we're going to see a cell that's going to grow longer, so it becomes very long and narrow, and part of the way that it grows is that the vacuole, which is a storage center inside of the plant cell. So you can see here the outline in gray of this vacuole. It's going to fill itself with water and solutes, and that can lead to the increased cell size. And you're going to see that this plant continues to get longer and longer because you also have more that will go through this process as those shoot apical meristems divide and then are going to lead to the next cells that enter the zone of cell elongation, as the ones that leave this zone then are going to differentiate. And this is just briefly looking at different types of plants. So when we look at shoot and leaf development, we're really only looking at vascular plants, and this is just looking at evolution in terms of time, different types of plants that we've seen. So your early vascular plants shown here, you have a shoot apical meristem that can divide in two, giving rise to new stems, right? You can see these in this direction, but you don't see any leaf structures. And so leaves and new branches, as we move over here, that's because over time you have a shoot apical meristem that then can give rise to a new type of meristem at the node of each branching off leaf, and these are known as axillary buds. These buds are meristems at the base of every leaf that can help give rise to a new branch. And if you've ever taken a cutting of a plant before, you might have seen a bud that forms, and that little bud, the axillary bud, if you cut it and put it into water, it will then give rise to an entire new plant system, so that you can start a new plant from that cutting. And actually you can see that really well here, right? This is what I was talking about, these axillary buds. Those are the ones if you cut a plant just right. So if you were to take a cutting, let's say you cut right here, and you put that top portion into water, you would find that new roots would come from that axillary bud in the water, and that would then lead to a new plant. And every time you have the new bud form, it would then give new branches, and you get a giant new plant from this really small cutting. We've already talked about the importance of flowers, and flowers actually are going to have their own stem cell, and it's going to be the floral meristem. This will give rise to all of the structures that make the flower what it is. And in order to develop, they need to have a signal, and this is a protein signal that will actually lead to the conversion of a apical meristem or an axillary bud into that floral meristem. And we all know what some of these signals are, right? Every spring you see flower development, so some of that relates to this process. So we already looked at one of our first hormones, florigen, and auxin we talked about previously. That's another hormone. So hormones are chemical signals that we'll talk about much more in human physiology later on. We get to the endocrine system. They influence many things, including physiology, development. In plants, they help to coordinate plant development, and they can also be important for responding to both internal and external environmental factors. We know that they can have a role in altering gene expression or cell division and expansion. Small amounts, and this is all of what I'm saying now about plant hormones is actually true for animals as well. Small amounts can have a huge effect, both locally and in faraway places. And the effect of one hormone oftentimes can be affected by another hormone. So there's going to be an interaction between hormones, and a really small number of them can help to shape the growth and development of plants. And this is also true for animals. So I thought we talked about auxin previously, but it's possible that we haven't. I've taught this course so many times, I can't recall. But anyway, we're going to talk about auxin now. We definitely talked about florigen. So auxin is, in fact, the first plant hormone that was ever discovered. It's important for growth. It promotes cell elongation in the shoots, and it actually is secreted by cells. in early developing leaves, and then it will help to connect that newly developed leaf to the vascular tissue that already exists in the plant so that you have a continuous vascular system. So it helps to guide this vascular differentiation so that it's continuous. And the way that it's going to work, if you look here at this picture, you have a developing leaf, so you have your shoot apical meristems, and then you can see this new leaf is developing here, so this is turning into a new leaf, and we're going to zoom in right on this little region in here and see what's going on in there. So if we zoom into this developing leaf, you're going to note that auxin is actually moving in one direction down through the plant from top to bottom, or from the developing region to the pre-existing region. So you can imagine it's being transported all the way from the tips down through here. And that becomes really important because it helps to connect the new plant growth there with the pre-existing vascular tissue. So I mentioned previously, auxin only moves in one direction. It moves downward to help to connect the vascular tissue, but we haven't said how that happens yet. So let's discuss that. So here you see auxin in blue entering into the cell. And when it enters the cell, it's actually uncharged. So this is an important point, and it's necessary that it's uncharged when it at first enters, because otherwise it wouldn't get through the cell membrane, because remember, the membrane is hydrophobic and will not allow for charged molecules to move across. But once it enters the cytoplasm, due to the pH of the cytoplasm, auxin then becomes negatively charged. I'm going to just underline that here. And then it means it can no longer diffuse across the membrane. So how does it get across? It has to have a channel. And these channels that allow it to move are known as pin proteins. And that pin protein is only found on the basal face of the cell. So plants or any cell, we talk about like the top portion versus the bottom portion, and it's all relative, but the bottom portion here we call basal. And that you can see is the only place where these pin channels are located. And that means that's the only way for auxin to get through. It has to go down in this direction. And then once it does that, it can become a neutral charge again, comes in, and then you have to start the whole thing all over again. And so you basically end up with this net movement in only one direction. And the result of all of this is it leads to the differentiation of cells to give rise to xylem and phloem. So you're connecting the vascular tissue of the new pre-existing plant to the new leaf. We'll look at this in class together. All right, so what we've been talking about so far is primary growth. That would be branching, right, new apical, new meristem, buds, formation, but the next thing we have to think about is secondary growth. So if you imagine as something is getting taller and taller and taller, it also gets wider and wider and wider, and you see this if you look outside, right? A small tree, for example, growing has a very thin stem, but as the tree gets larger and larger, the stem starts increasing in circumference as well as length. And that is important because it makes sense that you would have to support additional weight. So as something gets taller, it needs more width in order to sustain itself and not topple over. It also provides additional xylem and phloem tissue to enhance transport of both water and sugars throughout the plant. And the reason for this increase in circumference is due to other types of meristems we're about to discuss, and these are known as lateral meristems. So lateral meristems are found along the entire length of the stem, and after elongation is finished, they will start to form some of the different zones. So after the zone of elongation, as they start to differentiate, you'll start to see the formation of these lateral meristems. And again, it's going to produce growth in diameter, not length, and the number of these will increase over time in order to allow for this continued growth in terms of width. So we just defined lateral meristems, and now we're going to look at two different types of lateral meristems. We're going to have vascular cambion, one type of a lateral meristem, and then we're also going to have cork cambion as another type of meristem. And vascular cambion sounds like what it is. It is going to lead to new production of xylem and phloem. and we would call this the secondary xylem and the secondary phloem. And what you see as wood in a tree is actually the secondary xylem. In fact, secondary xylem is usually much bigger than secondary phloem. And when you look at the meristem, the vascular cambium on the inside, that will become the xylem, and on the outside will become the phloem. So let's see if we can zoom in on this picture here. So we're looking at the cross section, and now we're zooming in, and we're going to look for vascular cambium, which is shown in light green. So that, if I can make a little dot here, right, this light green right there, that circle that goes around, that's the vascular cambium. And then on the inside, you're going to see production of the secondary xylem. So if we now look at this next picture, as we zoom down here, because in this first picture, this only has primary. So you can see primary xylem here, and you can see primary phloem here on the outside. But now if we look at this picture, so again, we're going to find the vascular cambium, which is this light green right there. And then on the inside of this, we're going to see secondary xylem. So this is all this stuff here. And then secondary phloem is this darker green just outside. And you can see that even as this plant gets bigger and bigger, that the secondary xylem is really most of what you're seeing. So this wood, right, that's your secondary xylem. And a secondary phloem is like so tiny, it's almost impossible to see. It's going to be over here somewhere. And then you're also going to need to continue as this plant gets wider and wider. You imagine the outer layer keeps rupturing and having to rebuild itself because there's not enough surface area to cover the entire stem. And so the cork cambium, which is the next part we'll talk about, this is going to help to renew that outer layer as it ruptures, and the new layer will form. And so here you can see the cork cambium right in this inner portion, and that's going to help to continuously resupply the cork that will be produced, and that would be the bark on the outer layer. And the other thing you can see here that we actually could see it visually before, but now I'm just going to make the note that as this plant grows in diameter, as we're moving downward here, you can see that the secondary xylem, as it grows, is going to push the vascular cambium outward. So it always is going to stay in between the secondary xylem and the secondary phloem. So as the secondary xylem grows bigger and bigger and bigger, the vascular cambium will get pushed outward, and then you can see it's further out in this picture than it was in this initial picture. And it should make sense, right? Kind of intuitive, as same thing with the bark. As this circumference gets larger and larger and larger, obviously you have to add new cells to the vascular cambium. But again, vascular cambium is a type of meristem, so it's self-renewing, and it will keep being able to produce more of itself to take up that full space that it needs to as the diameter gets larger. And so when you look at the rings on a tree, it's just going to indicate a period of time in which there was growth and you had increased production of wood. So again, you're looking at all of the vascular cambium that was added over many, many years, and each ring is going to indicate a period of time in which the vascular cambium, the vascular cambium gave rise to the secondary phloem. So then this would be a period of time here. This narrow growth ring would be a period of time in which you had a very small amount of secondary xylem added, and then this larger growth ring would be a period in which you had more of the secondary xylem added in that period of time. We'll look at this question in class together. So we're going to look at root growth right now, and frankly, a lot of this is going to be analogous to what we spoke of for shoot growth. So just like we talked about before, you have the 3 zones of growth. So first you're going to have the zone of cell division, where the meristem is going to give rise to more tissue, and then those cells are going to undergo elongation, and then following elongation they're going to differentiate and mature. So very similar to what we saw before. So the root apical meristem, right, this will be analogous to the shoot apical meristem. Again, constant growth, same as we discussed before. Again, because they're not able to move, so this is the only way they can grow. Now, a few ways that roots are going to differ from shoots. So they're thinner, And that's important because they're involved in uptaking nutrients from the environment. So they have to have that high ratio of surface area to volume. And we know that the thinner something is, the more surface area relative to volume. So that's important. The other thing is that as they grow, instead of growing and encountering air, they're growing and encountering soil. You can imagine that that soil starts to eat away because it's rough at the tip of the root, which is a problem the shoots don't have. And for that reason, you need to have a structure known as the root cap, and that cap here will protect the meristems of the root so that they don't get destroyed. And then finally, you don't have any lateral structures on roots. There are no leaves that we're going to see appearing from the roots, so it's really just growing in length and branching. So this is just a nice chart that helps you to compare and contrast, and I'm showing you this one, but again, I recommend when studying that you do things like this on your own to help to look at similarities as well as difference. So roots we know are going to be thinner compared to stems. They have a root cap to protect. Stems do not. Lateral organs are not produced in the root. They are produced in the leaves, so that's again another difference. Primary growth is due to auxin. Absolutely, that's true for both. So this one is true for both. And secondary growth, yep, absolutely. Both of them are going to have increase in diameter, although we won't see it to the same extent for roots because we know they like to stay thinner. And branching, absolutely, in roots, they both are going to branch, but it's actually more extensive for the root system because, again, branching will allow for more uptake of nutrients. So roots can scatter like way out in the soil that you wouldn't see the same thing necessarily for a plant above ground, although it can vary. Some do branch more than others. So as I mentioned from the slide previously, auxin is going to have a very similar role as it did before in extending and connecting the vascular tissue. So just like we talked about before, it will allow for differentiation of new phloem and xylem to connect the new developing root with the rest of the root system. And same as before, we have secondary growth to increase diameter, add new xylem and phloem tissue. And again, that will be continuous so that you don't break this connection with the rest of the root system. And actually, while we're still on this page, I do want to highlight one thing because it's a nice cartoon image here, and I don't have it on the next slide. But if we zoom in on this central region here, right, so you can see that the xylem and the phloem are found inside the yellow portion on the outside, right? That's your endodermis. And then inside you're going to have the xylem and the phloem, and that's going to help to, when we talk about the next portion and we have new roots that are going to branch from this, we're going to note that new root structures are going to branch from inside here and then poke outward, and that's important so that you have continuity of the vascular tissue. So just pay attention to that in the next picture. I think that'll make more sense. So I just want you to focus on this central region here, and then we're going to go to the next slide. So we already know, again, roots have to branch extensively, and this picture here is why I wanted you to focus on the vascular tissue in the center. So note that in this central region, that's where the vascular tissue is. And again, I tried to draw this for you on the other slide, but I'm a really bad artist. So you can see that this new emerging root is growing from the vascular tissue, right? So this is extending from there. So you're going to have continuity, right? So you can see the continuity with the pre-existing vascular tissue. So that's really important because it's coming from within the endodermis so that everything, the endodermis will connect so that everything is connected to each other. And that's really all I wanted to point out there. And then you can see that the new root apical meristem is going to form because all new root structures are going to have that at the tip. So now we're going to be talking some more about plant hormones, and in particular how these hormones will bind to receptors, and that due to sensing your environment, these plants will then have a response to the hormones based on how they're sensing their environment. And one particular hormone that we've talked about before, auxin, is going to come into play here, and this is actually the discussion I was wondering before if we had talked about yet, and clearly we had not. So we're going to talk about responding to hormones and how small amounts can make a big difference, and also not just hormones, but how hormones can be triggered in response to environment. So plants can certainly sense their environment. You probably have seen this all the time, this tropism effect that we're going to talk about. We have seen, right, you put a plant on a windowsill exposed to sunlight, and where does it grow, right? Plants tend to grow toward the sun. And this effect we see quite frequently, and we're going to talk about how that actually happens. So tropism is just bending or turning in response to some signal. So we talked about it in terms of light. That's due to photoreceptors, and so the plant will sense sunlight and cause changes. So stems are positively phototropic. They bend toward the light. Again, that's what we were just talking about, the shoot system bending toward the light, whereas roots grow away from the light, right? They grow into the ground. We're going to focus more on this first one, so that's why I'm highlighting that one. That's the only one we're really going to talk about. But I'll mention the other ones briefly. So mechanical receptors, stems have negative gravitropic sensing, so they grow against gravity, where roots grow with gravity. And then you also have chemical receptors, and due to sensing chemicals, you have change in plants and how they function. But again, we can't talk about all of these, so I'm just going to focus on the positive phototropism, and we're going to jump into that right now as an example of how this works. So scientists were really curious about how plants grow and how the response to light occurs. So someone had an idea one day and said, if there is some signal in the shoot tip that is causing this to take place, because the tip is where you're growing from, how maybe could we figure out what the signal is? Maybe we could trap it. So what scientists did was they said, all right, well, what if I cut off the very tippy top of this growing plant and then place it on a gelatin block? And then whatever that chemical is, perhaps it will diffuse into that block over time. And then we can capture that chemical in this little block, put the block on top of the plant, and see what happens. So they did this. They did 2 experiments. One, they put the block directly centered on the tip of the plant. And in another experiment, they put it off to the side. And when they did this, they got 2 different results. When they put this straight on top of the plant, growth resumed straight upward. And when they put it off to the side, growth resumed, but you saw this tilt in the way it bent. And so scientists were trying to figure out what was going on. So sort of your punchline, we're going to get back to this and how this works in a minute, but a lot of this has to do with auxin, because that is your chemical, your hormone that helps with growth. It travels down the stem when it's been produced in the tip of the plant, and we're going to talk about how it can cause growth that can cause the plant to bend, and we'll discuss how that actually happens. So we don't have time to cover this in detail, but what I do want you to know is auxin production is partially stimulated by sunlight, and in addition, auxin can shift its location in the stem based on where the sunlight is coming from. So here we're going to say, again, in order to sense the sunlight, there have to be photoreceptors. So there are receptors in the plant that are going to absorb wavelengths of sunlight and then cause a response. And this response here, you can see, has to do with not only auxin production, but where it shifts in the plant. So if light is coming from straight above, then auxin production is equally distributed throughout the stem of the plant. So you can see here auxin shown in red is equally distributed in the stem. Now, if you see light is coming from the side, you still get auxin production, but you're going to note that auxin is shifted. So wherever the light is coming from, it causes auxin to basically move in the opposite direction. So auxin, in this case, if light's coming from the right, auxin gets shifted to the left. And then remember that auxin actually is causing growth of the plant. And so here, auxin's only causing growth on one side. And that elongation is going to cause the plant to bend. Because if you think about this, if you were to have, and the only way I can explain this, I mean, it's intuitive to me, but see if I can get you to think about this. If you have something that's straight, let's say you have this line here, and then you have this line here. It's sort of the straightest distance between 2 points is a straight line. So here's, right, so we have 2 points, and the shortest distance is a straight distance. So the minute you bend between those 2 points, you're increasing length. So auxin, by being shifted to one side causes increasing length on one side versus the other side. And so that means that you're going to have the outer region is going to be have additional length. It's the only way I can say it. The other way we can look at it is if you were to say this completed the circle. So let's say this arched all the way over, and then you would look at a smaller circle on the inside, which would be basically representing the outside of the plant versus the inside. This inner circumference is obviously smaller right here, so then you can see that by bending, this outer portion is actually bending because it's growing more versus this inner portion here that's not growing as much. We'll look at this in class together. And that's it. So you can see here again, different types of meristems. You should be responsible for that in terms of growth and development. Thinking about hormones and auxin, we talked about that quite a bit. We talked about that in terms of growth and continuation of vascular tissue, as well as the phototropic response. And I will see you eventually if the snow ever stops. Have a good one.